Happy to review with you the results of the trial I just presented 5 minutes ago. So this is a first in-human phase 1 clinical trial. So I think for this audience, you should take these results with caution in that this is a small data set, a first in-human study. With that as background, so small cell lung cancer, as you probably know, is a terrible disease, really unchanged in terms of clinically meaningful endpoints for patients with extensive stage disease, median survival 9 or 10 months from the time of diagnosis, and for recurrent disease, really only one approved drug, topotecan. So a lot of room for improvement here. What we were focusing on was a new target in small cell lung cancer, which we think may be a predictive biomarker for therapeutics, DLL3. This target is expressed in the large majority of small cell lung cancers and importantly is not expressed in any normal adult tissue. The drug that I presented on is called Rova-T or Rovalpituzumab Tesirine. This is an antibody drug conjugate that selectively kills DLL3 expressing cells. This again was a first in human clinical trial. There were 74 patients with small cell lung cancer enrolled in this trial. All had failed at least one standard therapy. All were treated with the investigational drug at doses shown here. We established a maximally tolerated dose and expanded cohorts at 0.2 milligram per kilogram given every 3 weeks and 0.3 milligram per kilogram given every 6 weeks. This study focused on safety and efficacy, and the efficacy data we really focus on the group of patients treated at what we consider to be an active dose range between 0.2 and 0.4 milligram per kilogram, where responses were observed. In a subset of patients, we had tumor available for testing for the target for this antibody drug conjugate for DLL3. So DLL3 was found to be detectable at some level in the large majority of patients, 88% of those in whom we had available tissue for analysis. I think importantly for this study, 2/3 of the patients, 67%, had what we're defining as high-level expression of the target, that is detectable expression by immunohistochemistry in at least 50% of the tumor cells. We reported that the toxicity of this agent was generally manageable. The most common severe toxicities were thrombocytopenia and a syndrome of serosal effusions, predominantly pleural effusions, which occurred in 11% of patients. We observed confirmed responses by RECIST criteria in 39% of the patients in the biomarker-selected patient population, DLL3 high, and an 18% overall response rate. Clinical benefit rate in the DLL3 high biomarker-selected population, 89%. That means an objective response or stable disease that was maintained over at least an interval of 2 scans. Patients on this trial were scanned every 6 weeks. Overall survival in this trial of previously treated metastatic small cell lung cancer, 32% in the DLL3 high patient population, which to us was a very encouraging signal, again with the caveat that these are early phase data. If we stack up what we saw in this trial against what would be the expected outcomes with the available standards of care, it looks something like this. So in the second-line setting, again, there's only one approved drug, topotecan. It has about a 17% response rate in the context of chemosensitive relapse disease. That's defined as disease that responds to chemotherapy and in which those responses are maintained for up to 90 days. In contrast to Rova-T, again, small numbers, but very high response rate here compared to the standard of care, and improvement numerically in the context of chemo-resistant and refractory disease with 1-year survival data that look promising, I would say, in both contexts. Even within the third-line setting where there is no FDA-approved therapy, there are reported retrospective series. This is a large series reported a couple of years ago a variety of drugs used in the third-line setting for small cell lung cancer with reported response rates retrospectively of about 18%. Small numbers in this trial in the third-line setting, but with Rova-T, we're seeing a 50% response rate and 33% survival at 1 year. Impressive numbers, again, with caution, small numbers, but these data really look promising to us as a novel therapeutic class for small cell lung cancer. So in conclusion, a new type of therapy for small cell lung cancer, an antibody-drug conjugate, biomarker selected. We demonstrate single-agent activity in the context of recurrent and refractory small cell lung cancer with comparable responses in the second and third-line settings. Responses and survival numerically improved versus historical controls. Again, take that with caution. The confidence intervals on these data are very wide. But importantly, I think most importantly, this is the first biomarker-directed therapy to be defined for the treatment of small cell lung cancer. And this is a biomarker that's expressed in the large majority of small cell lung cancers, so a biomarker that may be really of utility for the management of these patients. This drug at the defined MTDs, at the recommended phase 2 dose, has a manageable safety profile. And I think these results clearly justify further clinical development of this agent. We're currently enrolling to those studies. After meetings with the FDA, we've designed a study in the third-line setting, single-arm, with no comparator, because there is no approved drug in this space. That trial is called Trinity. That opened early in 2016 and is currently enrolling in multiple centers. And we have a number of studies that are on the launch pad, including a first-line trial in small cell lung cancer, a basket trial to look at other diseases in which the target DLL3 is expressed. And we're also exploring combinations with immuno-oncology agents that will probably launch later this year as well.